Our topic for today is WBC. Alright, last time we had completed with erythrocytes, RBCs. Okay, I hope everybody understood all the points in that lecture. See, all the components of blood. Okay, this part which is there in your textbook, it is a very theoretical part. It is not a conceptual part. So you will have to read all this 2 to 3 times so that you can remember many words. For example, last time we had studied words like polycythemia, like erythrocytopenia, correct? So all those words you will have to revise or you will have to write down so that you can remember them. Now in WBCs also we are going to get many such words. Okay, so today we are going to start with WBCs. Now WBCs are your white blood cells. Okay, and they are also called as leukocytes. Now what are WBCs? WBCs are basically the cells of your defense system. So they are basically protecting your body from foreign microorganisms, foreign bacteria, foreign viruses, all those foreign materials which can potentially harm your body, that is being undone by these cells. Okay, so these are basically the cells of your defense system. Okay, so let us start with the structure. Okay, just like we had studied the structure of RBCs. So the structure of leukocytes or WBCs, the first thing is that these cells do not have any color. Okay, RBCs are red in color because of hemoglobin. However, these cells, these WBCs, they are colorless. Are you getting my point? Okay, so they are colorless. The second thing is, if we want to talk about the shape, then the shape of WBCs, there is no particular definite shape. Okay, if you talk about the shape of RBCs, you can say that they are biconcave. But WBCs do not have a standard shape, so they are amoeboid. Okay, what is the meaning of amoeboid? Amoeboid means just like an amoeba doesn't have a fixed shape, WBCs may differ in shapes. See, basically there are many different types of WBCs. Okay, so each WBC will have its own shape, so that is why we use the word amoeboid, no fixed shape. The third thing is that these cells are nucleated. Okay, RBCs are enucleated or non-nucleated. That means they do not have nuclei, but WBCs have a nucleus. In fact, different type of WBCs show different type of nucleus, and on the basis of nucleus also we can differentiate WBCs. We'll come to that later on. So are these 3 points clear? Okay, this is about the appearance. Coming to the total WBC count, do you remember the total RBC count? So in your textbook, the RBC count for males it was between 5.1 to 5.8 million, and for females it was 4.3 to 5.2 million per millimeter cube. Now compared to the RBC, the total WBC count is much less. Okay, you do not require WBCs in such high amounts. So if you want to write about the total WBC count, then in both males and females, so again the WBC count doesn't differ much in males and females like it does in case of RBCs. WBCs, both males and females will show almost the same number of WBCs, and that count is how much? It is just 5,000 to 11,000 cells per millimeter cube. That is it. Compare that to RBC, this was what? 5.1 to 5.8 million. So the number, the total count of WBCs is much lower as compared to RBCs. Okay, so this number is there for both the sexes, males as well as females. So I hope this is clear. Okay, now we come to both the conditions where what happens if this count or if this number goes above this value or if the number goes below this value. So once again, if the number goes above this normal value, if the WBCs present in your body are above the normal range, then the condition is called as, so if the count is above 11,000, then the condition is called as leukocytosis. What happened? What was the word that we used when RBC count was above the total normal count? The word which we used was polycythemia. Now this word and that word, these are both totally different words. They have got no relation. Okay, so please remember, leukocytosis means the number of WBCs is above the normal range. Okay, so I will write down in bracket WBC number goes up. Similarly, what happens when the WBC count is below the normal range? If it is below 5000, then the condition is called as leukopenia. Now, this word is repeating itself. Remember, erythrocytopenia, and I told you penia means the number is below the normal range. So, if you remember erythrocytopenia, then leukopenia is easy. Leuco is leukocytes. Penia is reduction in number. So, this word matches with erythrocytopenia. Is this clear? So, this is when the WBC numbers goes below the normal count. Is this clear? Okay. Now, when does leukocytosis happen? When does leukopenia happen? What do you think? When will the WBC number be above the normal count? So whenever you have an infection, okay, you have a viral infection, you have a microbial infection, you have a bacterial infection, कोई भी infection के case में your body will have to fight back. So it will produce more WBCs. So at that time during infection, your WBC number will be more than this normal count. Are you getting me? So during infection, your WBC count increases above the normal count, and that condition is called leukocytosis. Is this clear? When will leukopenia happen? WBC number कम कब होगा? So there are certain disorders where the WBC number goes below the normal count during infection. For example, you must have heard of a disorder called AIDS. AIDS मतलब acquired immunodeficiency syndrome, and the virus which causes AIDS, it is HIV, human immunodeficiency virus. So when this virus infects the body, what it does is, यहाँ पे नाम क्या है, acquired immunodeficiency syndrome, मतलब immune system जो है वो deficient हो जाती है, immune system becomes weak. So how does this virus make the immune system weak? Basically it goes and attacks your defenses. Now in WBCs there are a special type of defense cells in our body called as Cd4 cells. Now this we will be not learning in your 12th standard. This is extra information. All those who are doing NEET, they should know this. ठीक है, आप NEET नहीं भी कर रहे हो तो भी आप जान सकते हो. So these cells which are Cd4 cells, okay, they are a type of lymphocytes. Now lymphocytes will be doing in today's lecture. एक type के WBC है lymphocytes, और वो lymphocytes बहुत type के हैं. One of the type of lymphocytes is this Cd4 cell. So it is a lymphocyte. And what is a lymphocyte? A lymphocyte is a type of WBC. So these are the chief defense cells. Now what this virus does is it directly attacks the Cd4 cells and it begins to destroy them. So your Cd4 cells number begins to decrease in the body, and because of that your immune system becomes weak. It becomes so weak कि common cold जैसे infection से भी आपका immune system fight नहीं कर सकता. तो in conditions or in disorders like AIDS, or another example of disorder is tuberculosis, TB. So AIDS या TB जब होता है, at that time what happens is your WBC number goes lower than the normal count, and that condition is called leukopenia. Is this clear? Okay. So cytosis means above the normal value. Penia means below the lower value. Okay. Now in your book they have not given this. Formation of WBCs is called as, so it is called as what? What is formation of RBCs called as? Erythropoiesis. Similarly, formation of WBCs can be called as leukopoiesis. Now this is not given in the book, but obviously they should have given it. So please remember, leukopoiesis means process of formation of WBCs. Is this clear, guys? Okay. So we have basically started with our WBCs. I have just given you a summary of WBCs. We have to study about different types. So up till now, what all we have learned? We have learned about the shape and structure. Okay. What are the important points? First of all, it is nucleated. That means nucleus is present. Colorless it is, not red. And the third thing is, it is amoeboid. And one more important point, since I have written here amoeboid, it can undergo diapedesis. I have already explained what is diapedesis. Diapedesis means it can pass through the fenestrae of capillaries. Okay, so I will explain again if you don't remember. This is your capillary. These are the capillary pores, or these are the fenestrae. So if this is your WBC, it can squeeze through these pores. So it will change its shape and it will squeeze through. So if there is an infection in this area, the WBC which is passing through the capillaries, it can come out of the capillaries. And this movement of WBCs passing outside the capillaries is called diapedesis. It is only possible because WBCs do not have a fixed shape. They can change their shape. Is this clear? Okay. So this is about the structure and shape. Then we learned about the WBC count, much lower compared to RBC, 5000 to 11,000 cells per millimeter cube. Then leukocytosis when the number goes above the normal count, leukopenia when the number goes below the normal count, and what is leukopoiesis? This is not given. Okay, when is this happening? This is happening in disorders like AIDS or TB. This is happening, this is happening in case of AIDS or TB, the number will go down and it will increase during any normal infection. You are having a fever, you are having any bacterial infection at that time, the number of WBCs will be high. Now there are certain cases where the WBC count goes very high. For example, some people might show a WBC count of 1 lakh to 4 lakh. Now look at this range, 1 lakh to 4 lakh cells per millimeter cube. This is such a huge number. Normal count is 5000 to 11,000. See, in leukocytosis also the count is not very high. It may be around 15,000, 20,000, 25,000. But here if you see, the count is above 1 lakh. It is from 1 lakh to 4 lakhs. Such extreme conditions, for such extreme cases where the WBC number is 20 to 40 times higher than the normal count, this condition is called as leukemia. Now, first of all, in your book, the spelling for leukemia given is Leukemia. Now, this is the American spelling. This is the British spelling. Which English do we follow? We follow British English. So we should be writing this spelling, not this spelling. Please remember this. So if I am adding A in front, that means this is British spelling, this is American. In your book, this is given. It doesn't make much of a difference, but this is ideally the spelling that you should write AE. Now leukemia is also called as blood cancer. Okay. So a common person will say कि blood cancer वो patient को है. Blood cancer means what? The WBC number is so high. What is cancer? Unlimited multiplication of cells is called as cancer. So blood cancer में WBCs, RBCs do not multiply. ना blood cancer में. In blood cancer, the WBC count is so high. Look at the normal count. Look at this count. Even this count is not as high as this. So what is the difference between leukocytosis and leukemia? Leukocytosis is rise in the normal count, but not as much as this value. This is to rise by a huge margin. आप समझ रहे हो ये? So ये condition को बोलते हैं leukemia. So leukocytosis, leukopenia, leukemia, all these words are very clear. Okay, and finally leukopoiesis. So with that we have completed the general introduction of WBCs. Now we have to start with the most important part in WBCs, and that is about the types of WBCs, because this is what is normally asked in the papers. Again I am repeating, this is a theoretical topic. It is not a conceptual topic. Okay? You can remember the meaning of the words, but if you can't remember the words during the exam, you will lose on marks. So guys, please, if you want, write down these important words. Okay, make a chart if you want, or read them 2 to 3 times. Do whatever you want, but those words should be ingrained in your memory. आपके brain के अंदर वो याद रहने चाहिए. समझ रहे हो आप ये? ठीक है. Now, WBCs are basically called as polymorphic. Let me write down this word now. What is the meaning of polymorphic? Poly is many and morphic is shape or structure. So WBCs have different shapes and structures. All the WBCs do not have a common shape and structure. That is why they are called polymorphic. Erythrocytes are not polymorphic. All the erythrocytes are red in color, they are biconcave, they are non-nucleated. So they are not polymorphic. WBCs are polymorphic. Ye clear hai? Now, the two main types of WBCs are First group of WBCs is called as granulocytes, and the second group of WBCs is called as granulocytes. Now this is going to become very theoretical. Please pay attention. If you want, you can pause the video. Make sure you have understood whatever I have taught up till now, then go ahead. Okay, and again, please make it a habit to write down important points in your book. Okay, so that you don't get confused with the lecture. Now, what are granulocytes? What are agranulocytes? First of all, out of the total WBCs present in your body, granulocytes form 72%. So out of all the WBCs present, 72% of WBCs are granulocytes. Agranulocytes are how much? They are 28%. Is this clear? I am just talking about WBCs. ऐसे नहीं बोल रहा हूँ जितने भी ब्लड सेल्स है उसमें से 72% नहीं, सिर्फ WBCs में से ये 72, ये 28. Now, first of all, why these names? Once again, I will repeat. Cytes mean cells. Okay, erythrocytes, leukocytes. So cytes in blood terminology means cells. Granulocytes are the WBCs which show a granular cytoplasm. Now see, if I am just drawing a general structure of granulocyte, then this is the shape of the WBC. Alright, it will have a nucleus. So just suppose this is the nucleus. Correct? I will explain why this nucleus I have drawn like this. But the most important thing is these cells will have pigments in the cytoplasm. Can you see these red colored pigments which I am drawing? Okay, these are called as granules, or they are also called as pigments. So basically the cytoplasm of granulocytes have granules or pigments in them. That is why these are called as granulocytes. Whereas over here, if you see, then in these WBCs, the granules will not be present. So if this is the WBC, which is an agranulocyte, then it will have a nucleus. It will not show any pigments or granules in the cytoplasm. So it is a granulocyte. Simple. Granule present, granule absent. So let me write down the second point over here. 72% के नीचे I will write cytoplasm is granular. Or it is pigmented. Whereas over here, what should I write? Cytoplasm is agranular, or granules are absent. So it is always very important to understand the meaning of the words in biology so that it becomes easier to remember. If you just mug up these words, then you will not know why this is agranulo, why this is granulo. सिंपल है, अग्रैन्युलो मतलब ग्रैन्यूल्स नहीं है, ग्रैन्युलो मतलब ग्रैन्यूल्स है. कहाँ पे है? साइटोप्लाज्म में. Now coming to the next point of difference between these two groups of WBCs. Now coming to the nucleus. So the nucleus basically is multi-lobed in the case of granulocytes. So a single nucleus will have many lobes. For example, if I draw the structure over here, then if I say that a nucleus is trilobed, so that means the nucleus will be like this. So can you see 1, 2 and 3 lobes? If I say that the nucleus is bilobed, okay, then the nucleus will be like this. If I say that it is pentalobed, then it will be like this. Are you getting my point? So basically over here, a single nucleus is having many lobes. The number of lobes may be between 2 to 7. Okay, so that I am just giving you an idea. So basically these WBCs have a multi-lobed nucleus. Is this clear? And since, see, all the granulocytes which are present, they are again, these granulocytes are divided into 3 groups. So each group will show different number of lobes. So that is why the granulocytes are also called as PMN cells. Now what is the meaning of PMN? PMN stands for poly-morpho-nuclear cells, or you can also write down leukocytes. See, so many words. Polymorphone-nuclear means there are many different types of nuclei. Some may be bilobed, some may be trilobed, some may be tetralobed, pentalobed, hexalobed, heptalobed. So there is no one particular structure of nucleus, even in the granulocytes. Okay, we will be studying about that. So that is why these are called polymorpho. Poly is many, morpho is shape, structure. Nucleus is many different nuclei are present, no fixed structure. Is this clear? So these are called PMN cells. Over here, if you see, then the cells have a single nucleus. They have a large nucleus. So I will write down single large nucleus is present. Now this point which I am writing down, they are very important for you because a question can be asked, differentiate between. Okay. So in that case, these points will be important. And the other thing is, in your book, they are not given in this comparative form. If you read the textbook, you will be able to find these points, but as a comparison wise नहीं दिया. Is this clear? So अभी तक कौन से points किया हम लोग ने? Granulocytes में cytoplasm is granular, यहाँ पे agranular or absent. Here nucleus is multi-lobed, having more than one lobes. Here single large nucleus, one single nucleus is present. 72% of WBCs, 28% of WBCs. What other point do you think I should write? Most important point, leucopoiesis. What is leucopoiesis? Formation of WBCs. WBCs are formed from what? WBCs are formed from stem cells. Please remember. RBCs are formed from where? So RBCs in the case of fetus are found in liver and spleen, and in the case of adults they are present in red bone marrow. WBCs are formed from which cells? Stem cells. What are stem cells? Stem cells are special cells which can grow into any organ or tissue. Okay. Now again, stem cells are of two types. Okay. So I hope you are not getting confused. If you think that my pace is fast, you can again rewind the lecture, again go back to this part and again start, or you can even change the speed. Okay, there is a play speed option in YouTube, so you can reduce the play speed to 0.75 or 0.5. Anyway, stem cells are again of two types. Okay, so I will use a different color. There are stem cells which are called as myeloid, Myeloid. Myeloid stem cells, and the other type of stem cells are called as lymphoid. Now you don't have this in your syllabus. You have these words but you don't have this. So let us not go into the detail because it will become very confusing. So just remember, stem cells which are there in your body, they are of two types, myeloid and lymphoid. Now why do you need to remember this? Because these cells are formed from myeloid stem cells, whereas these WBCs are formed from lymphoid. Is this clear? So these granulocytes are formed from one type of stem cells which are called as myeloid stem cells. So next point I am going to write down over here. Let me see if it will be visible on the screen or not. Okay, it will be visible. So the next point is, these are produced from myeloid, myeloid, myeloid stem cells, whereas these WBCs are produced from lymphoid stem cells. Alright, so don't think too much about what is the difference, just remember this. Okay, so one last point in both these cases. Now what is the difference between granulocytes and agranulocytes apart from these points? See, these are formed from lymphoid, these are formed from myeloid stem cells, these are formed from lymphoid stem cells. But what happens after they are formed? So once the granulocytes are formed from these myeloid stem cells, so obviously through cell division, through mitosis, they will be formed because these have a nucleus. So WBCs will be formed by mitosis. But what happens is, once these granulocytes are formed, they will not undergo further division. आप समझ रहे हो क्या? For example, अगर आपके पास यहाँ पे माइलॉइड स्टेम सेल है और ये माइटोसिस के बाद फॉर्म कर रहा है ग्रैन्युलोसाइट्स, ओके, it is forming many granulocytes, but once these granulocytes are formed, then there is no further division. ऐसे नहीं होगा कि ये ग्रैन्युलोसाइट्स माइटोसिस करके और ग्रैन्युलोसाइट्स फॉर्म करोगे. प्लीज रिमेंबर, so the division stops after the cells are formed. So once the granulocytes are formed from these cells, there is no further division. Whereas over here, after the agranulocytes are formed from lymphoid stem cells, these will still continue to multiply by cell division. So यहाँ पे after granulocytes are formed, cell division will stop. यहाँ पे after the agranulocytes are formed, cell division will further continue. Is this clear? Are you getting my point? So what should I do? I will write down the point. कहाँ पे लिखूँ? Should I rub this 72% off and write it over here? Okay, if you want, I will write in bracket 72% and this is 28%. Now over here I will write down granulocytes do not undergo further cell division. So once they are formed from myeloid stem cells, cell division will stop. So this is somewhat like meiosis. Meiosis 1, meiosis 2 होने के बाद आगे division नहीं होता है. Similarly यहाँ पे, but this is not meiosis. Here what should I write down? Granulocytes, okay, undergo further division after being formed. Is this clear, guys? So these are all the differences, okay? These are all the differences between these two types of WBCs. So once again, let us recap. What are the two types of leukocytes? Granulocytes is one category, agranulocytes is the second category. First point for each of them, what will you write? Granulocytes have granules in the cytoplasm, correct? They are pigments. That is why these cells are called granulocytes. These do not have granules or pigments in their cytoplasm. They are called agranulocytes. Second point is about the nucleus. Here the nucleus is polymorpho-nucleus. So there are many different types of nuclei and they are always multi-lobed. Okay, so bilobe, trilobe, tetralobe, and all the different cells will show different nucleus. Here the nucleus is only single and large. So these are not called polymorpho. Polymorpho matlab many different types. Okay, then we come to the next part, which is myeloid stem cells. So these granulocytes are basically formed from the myeloid stem cells. Okay, and these agranulocytes are formed from lymphoid stem cells. Another difference, these cells once they are formed, they cannot undergo further cell division, they cannot undergo further mitosis. These cells when they are formed, they can undergo further cell division or further mitosis. Is this clear? And finally 72%, 28%. Now still we have not come to the most important part of WBCs. We have to study the types of granulocytes. See, if you are watching this for the first time, I am sure this was not taught to you in school. You must be thinking, कितना division है ये? पहले तो you studied blood. Then blood is divided into plasma and formed components or cells. Then formed components में RBC erythrocytes, WBC leukocytes, thrombocytes, platelets. RBC में तो कुछ नहीं. WBC में दो टाइप, granulo, agranulo. अभी ये granulocytes में भी और तीन टाइप्स है. Okay, so depending upon the type of granules present, the granulocytes are further divided into, okay, first is, so what I will do is I will just use a different color to write down neutrophils. Then the second type of granulocytes are Basophils. And the third type of granulocytes are Eosinophils, or they are also called as Acidophils. Now, why are there three types of granulocytes? First of all of them is going to have a different type of nucleus. Second is their function is going to differ. And the third thing is they are, if you look at the words, this is neutral, this is basic, this is acidic. So the granules, if you want to observe these different types under the compound microscope, तो आपको stain करना पड़ेगा ना? Why? Because WBCs are colorless. So how will you recognize which type of granulocyte it is? So you have to add a stain. Stain मतलब क्या? Stain is a dye which will help you to observe the cell under the microscope. So these cells, they can be stained with neutral dyes. These cells, they can be stained with basic dyes. And these cells can be stained with acidic dyes. Is this clear? So our next part is going to be, first we are going to study this, then we are going to study this, and then we are going to study this. Now, I am not going to cover agranulocytes in this lecture. There are again two types of agranulocytes. First is monocytes, second is lymphocytes. So if you want, I will write down over here. Okay, two types of agranulocytes are, first is lymphocytes and second is monocytes, but that I am not going to cover today because it will become very confusing for you. This is a very small part, still I will teach you that part in the next lecture. Is this clear? So three types of granulocytes are given over here. This is the first, alright, this is the second, and this is the third. Is this clear? So next part we are going to start is on neutrophils. We are going to start in this lecture itself. I am going to teach you that. If you want, you can take a short break. If you want, you can write this down in your notes. Okay, you can pause the video and then we will resume with neutrophils, then basophils, then eosinophils. Okay. Okay, so should we start with the next part? So we are going to first start with neutrophils, then we are going to start with basophil and then eosinophils. Okay, so I will just rub this off. Hopefully people have copied whatever they needed. So the questions which are asked from this part is, first of all, differentiate between granulocytes and agranulocytes, right? Then write a short note on WBCs. So if a short note is asked, then you have to add all the words like leukocytosis, leukopenia, okay? Then you have to add leukemia, then total WBC count, what is diapedesis, what is the structure, so all that you have to add. But most of the questions are basically asked on these types. So if they ask you write a short note on types of granulocytes, you have to explain neutrophil, basophil, eosinophil. Now let us start with the first type. So the first type of granulocyte that we will be studying, they are called as neutrophils. So what should I do? So this will be a comparative diagrams and points I will be writing. So you can compare that at the end. So let us make one big chart. So the heading will be what? Heading will be granulocytes. Okay, is this visible in your video? Okay, it's just visible. I think it's good if it's, I will just, alright. Now granulocytes are of three types. The first type that we will be studying, okay, that first type is called as neutrophil. So I am writing it down over here, neutrophil. Alright. The second type that we will be studying, that is called as the basophil. Okay, basophil. And the third and the last type we will be studying is called as the eosinophil, or it is also called as acidophil. I am not writing it down here. So I hope this is visible. Yes, it is very clearly visible. And now we are going to write down the theory. First of all, let me draw the diagrams. Okay. The first diagram I am going to draw is for neutrophil, so I am going to use a blue colored ink. Okay. Now, the first thing in neutrophils are, these are the largest among all the granulocytes. Okay, and also number wise also they are maximum. So neutrophils take up around 70% of the total WBCs. Total WBCs, huh? So granulocytes, granulocytes, all the WBCs together, among them neutrophils are 70%. Okay. Also I will write down these numbers. Basophils are approximately 0.5 to 1% of the total WBCs. Please remember, I am comparing these numbers with total WBC count, not 0.5 to 1% of granulocytes. नहीं, ऐसा नहीं है. And similarly, eosinophils will be 1 to 3%. So total this plus this will be 72%. Clear? Okay. Now let us start with neutrophil. So now the neutrophil is one of the largest cells. In fact, among the granulocytes, it is the largest. So what I am going to draw is, I am going to draw this cell. This is the neutrophil. Now we come to the granules which are present. Now the granules are very large in size. Okay. So what I am going to do is, I am going to use a green pen. Doesn't mean that the granules are green in color. Okay. So there are many granules which are present and they are very small. They are very fine. Okay. So very fine granules, but they are evenly distributed. मतलब साइटोप्लाज्म में सब जगह पे वो प्रेजेंट है. Coming to the nucleus, the nucleus has many lobes. Okay, the lobes may be between 2 to 7. So if you see the diagram given in your book, they have shown a 3-lobe structure. You can draw 4-lobe, 5-lobe. 2 से 7 के बीच में lobes होने चाहिए. I am going to draw 2-lobe structure. So the nucleus is somewhat like this: 1 lobe, 2nd lobe, 3rd lobe. Is this clear? So this is the tri-lobe nucleus present. Again, I will finish drawing the granules. The granules are very fine. Fine का meaning है they are very small, but they are evenly distributed. So देखो, it is very simple to remember. If the size of the granules is smaller, then their number will be more. Suppose if the size of the granule is this much, then the size will be bigger but the number will be less. Common sense hai? Okay, so this is your diagram for neutrophils. Now what do you need to remember in neutrophils? First of all, 70% of total WBCs are neutrophils. What are neutrophils? They are a type of granulocyte. Now second point is coming to the nucleus. The nucleus is polymorpho-nucleus. So all these are polymorpho-nucleus having 2 to 7 lobes. So the range is given. So एक न्यूट्रोफिल में 2 लोब है तो दूसरे में 3 भी हो सकते हैं, 4 भी हो सकते हैं, 7 तक कितने भी हो सकते हैं. So this is very important to remember. Next point. What are the functions of this neutrophil? Very important. You have to remember the functions of all these cells. These are basically phagocytic in nature. Have you heard this word? Phagocytic. Phagocytosis. You can say that the neutrophils, they help in phagocytosis. What is phagocytosis? Basically, whatever microbes are present, these microbes will be eaten by this cell. So what is going to happen is, these are already amoeboid. Okay, so I will write down, these are amoeboid. Amoeboid means they can change the shape of their cell. So how will they move? Just like an amoeba, by adjusting their cell membrane. So they show amoeboid movement. Okay. Now coming to this phagocytic, or they show phagocytosis. Very simple. Just imagine this is the bacteria which is present, and this is the neutrophil. How will this neutrophil eat this bacteria, or how will it undergo phagocytosis? Very simple. The shape of the cell membrane will change. Now the shape will become like this. Then what will be the change of the shape? Then the next shape will be like this, and then it is going to eat away the cell, so the shape will become like this. Now this foreign microorganism has been eaten and it will be digested by this cell. This entire process is called as phagocytosis. Very simple. So what is the function of neutrophils? Neutrophils have only one function, and that is called as phagocytosis. These are phagocytic cells. That means they can eat the foreign microbes. Is this clear? Okay. So again, let me draw the original diagram. Okay. Now we have not written about the granules, so let me write about the granules. Granules are fine. Fine means they are very small, and hence I will write down in bracket more in number. Is this clear? Okay. Most important point, why is the name neutrophil? Neutrophil means these pigments or these granules which are present, these granules can be stained by neutral dyes. Very important point. सबसे important point यही है कि these pigments which are there, these are normally colorless, but if you want to see them, then you can add a neutral dye and these become colored. So I will write down over here that granules are stained by neutral dyes. Is this clear? Okay. Now, one more thing which I forgot to tell you. What is the function of these granules? So, granules are present here, over here, and also over here. So, basically, these granules which are present, they are secretory cells. Should I write down the word over here? Secretory. Secretory मतलब cells जो कुछ secrete करेंगे. So these secretory cells, they can secrete करना मतलब release करना. So these granules, they can secrete enzymes or they can secrete proteins or hormones. So depending upon the type of granule which is present, these granules will secrete different things, and that is why these granules are secretory cells. We will see what these what is the meaning of this word in this example and this example. यहाँ पे तो कुछ secretory cell का काम नहीं. Okay. So now, is this neutrophil clear? Okay. Now, have you heard of the word pus? P-U-S. सुना है क्या ये word? Pus सुना हुआ है? You know that whitish liquid which is discharged normally after an injury. So there is inflammation, then there is swelling, then pus निकलता है. What is pus? So pus is basically a mixture of the neutrophils which are dead plus the foreign microorganisms which have been killed by the neutrophil and plus whatever tissue which is damaged. So suppose if you have some damage over here and यहाँ से pus निकल रहा है, तो वो pus के अंदर क्या क्या रहेगा? वहाँ के neutrophils which are already dead plus वो microorganisms जो भी मर गए हैं और उसके साथ इधर का जो आपका टिश्यू डैमेज हुआ है वो भी. सो ऑल दीज थ्री थिंग्स कंबाइंड विल फॉर्म दैट व्हाइटिश येलोइश कलर्ड लिक्विड व्हिच इज कॉल्ड एज पस. सो पस के अंदर क्या रहते हैं? आपके न्यूट्रोफिल्स रहते हैं. अब समझ रहे हो, सब कुछ मरा हुआ रहता है. न्यूट्रोफिल्स आर डेड, माइक्रोब्स आर डेड एंड टिश्यू डैमेज सेल्स आर आल्सो डेड. इज दिस क्लियर? सो दिस इज गिवन इन द बुक. सो इज न्यूट्रोफिल्स क्लियर? Okay, remember the lobes are between 2 to 7. Diagram given in your book is showing 3 lobes, so I have drawn this. Remember, granules are the smallest, fine granules, evenly distributed. The entire cell is the largest among the granulocytes. Okay, then amoeboid movement दिखाते हैं, so they can undergo diapedesis. What is the function? Phagocytosis. Is this clear? Okay, and they are stained by neutral dye. If you want, this point should be written first, क्योंकि ये नाम जो है वो इसके ऊपर से. So if you want to see this granulocytes, if you want to see the neutrophils under a microscope, you will have to use a neutral dye. Is this clear? Okay, नंबर याद रखना है, ये 70% है, 72% में से ये 70% है. Next, coming to the next type of cell, which is your basophil. So basophils are slightly smaller compared to the neutrophil. So I'm going to draw a smaller cell. All right. The other thing is that the basophil is having large size granules. Okay. So यहाँ पे granules are fine, smaller in size. यहाँ पे they are larger. So I am drawing these granules over here. And because they are larger in size, their number will be lesser. So I hope everybody understands this now. So these are the granules which are larger in size. And the most important thing is the nucleus. The nucleus is irregular, मतलब ऐसे कोई particular shape नहीं है. It may be lobed, it may not be lobed. It is normally irregular, so you can show a twisted nucleus if you want. Okay, so that is the nucleus, and this is your basophil. Now, what are the points that I am going to write? How much is the proportion of basophils compared to your WBCs? they just form 0.5 to 1%. So ये सबसे कम टाइप के WBCs है, okay, proportion wise. So that is that. Next, nucleus if you want to write down, then irregular nucleus, or you can write twisted or folded nucleus. Then coming to the function, okay, anyway, before we write down about the function, let us write down about the granules. So granules are larger in size, correct, and because they are larger in size, they are lesser in number. Very simple. Coming to the next point, which is why they are called as basophil, they are stained. ये जो granules है, these granules are stained by basic dyes. Okay, so I will write down granules are stained by basic dyes, and they have given an example of basic dye. Okay, so this is basically a blue colored dye which is called as methylene blue. Visible है क्या ये? So this is methylene blue. Methylene blue you must have seen in your lab जब भी 11th standard में थे, blue color का dye रहता है. So if you use this dye, this is basic. Basic मतलब क्या? pH is more than 7. So अगर आप ये यूज करते हो तो ये granules सब blue color के stain हो जाएंगे. So basic dye stain करता है, that is why they are called basophils. Clear है? Coming to the most important part, function. Now ये वाले जो cells है, ये है phagocytic. These cells are non-phagocytic. Please remember, these basophils will not undergo phagocytosis. So I will write down. We don't have much space over here. I will write down over here. These are non-phagocytic. So काम क्या है फिर? These cells are released during allergic conditions. Allergic conditions मतलब cold, cough, ठीक है, itching, all the sudden things which develop allergic condition के time पे ये release होते हैं. Okay, and what is the function of these cells? These cells, they will release 3 types of substances. See, इसमें बहुत कुछ याद रखना है basophils में. Please remember. ऐसा नहीं है कि basophils is very easy to remember. First of all, irregular nucleus, clear. Granules larger in size, clear. Stained by basic dyes. Basic dyes का example है methylene blue, so I have shown this blue in color. All right. Unfortunately, I didn't have a black pen. Otherwise, I would have used a black pen for this. So समझता. Anyway, coming to the function, non-phagocytic है. ये phagocytosis नहीं करेगा. ये microbe को नहीं खाएगा. What is the function? They are released during allergic conditions, and basically these cells release 3 substances. Okay, and I am going to write down the names of these substances in green color. First thing that these cells release, it is called as heparin, Heparin. Second thing which is released is called as histamine, and third thing which is released is called as serotonin. So you have to remember, अभी ये तीनों के functions आपके book में नहीं दिए हैं, but for NEET entrance exam it is important for you to know the functions, or in general also it is important. Heparin is an anticoagulant. What is anticoagulant? See, the blood which is present inside your body, it is in a liquid state, but if you have a tissue injury, then that blood comes out and then it solidifies, correct? If you have some damage to the skin, then what happens? Blood will come out, but will it remain liquid? No, it will become solidified. So that is called coagulation. तो बॉडी के बाहर क्यों कोएगुलेट हो रहा है, बॉडी के अंदर why it is not coagulating? Because inside the body the blood is having this chemical which is heparin. Now when the blood is exposed to outside air, this does not work. Okay, so this heparin is also called anticoagulant, so it keeps the blood in the liquid form. Is this clear? Next, what is histamine? So these cells are released in allergic conditions. During allergy, histamine is released in lot of quantities. Okay, आपको जब cold होता है तो आपको histamine release होता है, and because of that excessive histamine, what happens is your nose becomes blocked. You have a lot of mucus which is released. So all the things which happen during allergic conditions are because of histamine. So histamine तो release होने ही वाला है, allergy के time पे ये release होगा. And what is serotonin? See, serotonin has got many functions, but you can call it as a feel good hormone. Feel good hormone मतलब whenever serotonin levels in your body increase, you have a good feeling, मतलब feeling of well-being, सब कुछ अच्छा हो रहा है, आप समझ रहे हो? So this is that hormone or this is the neurotransmitter. So three things which are released by the basophils: heparin, anticoagulant; histamine, which is released during allergic conditions; and serotonin, which is a feel good hormone. Is this clear? So I hope basophil is clear. Differences between this and this: first is nucleus, second is size of the granules, third is staining of the granules, neutral dye, basic dye, and these three things are. So now you realize secretory cells क्यों बोलते हैं? ये जो cells है आपके, ये तीनों चीज secrete कर रहे हैं. These granules are secreting these three things. Coming to the last type of granulocyte, which is called as eosinophil. Okay, so again now eosinophil, they are slightly larger compared to basophil. Okay, so you can draw eosinophil like this. Coming to the nucleus, it is bilobed. Please remember, दो ही lobes present है. So if you see the diagram given in your textbook, again you will see that there are two lobes. And coming to the granules, नाम से Eosin और the other name is Acidophil. So acidic dye है, they are stained by red colored dyes. So these are the granule pigments that you can see. Okay, now again proportion, total WBC में से कितने परसेंटेज eosinophil है? 1 to 3%. So देखो, this plus this is how much? 72. So I should write over here granulocytes are 72%. Is this clear? Now coming to the first part, which is nucleus, what should I write? Over here I will write down bi-lobed nucleus. Second thing is write about the granules. So I will write down granules stained by acidic dye. Acidic dye मतलब जिसका pH होगा less than 7. And the best example of acidic dye, again you might have seen this in your laboratory, it is called as Eosin. तो Eosin करके भी एक dye रहता है, जैसे यहाँ पे है methylene blue, वैसे over here I will write down the example of acidic dye Eosin. That is why the name of these are eosinophil. So अगर आपको ये colorless WBCs को देखने हैं, तो you will have to add this red pink colored dye which is called eosinophil. Okay. Next, function is, these are again phagocytic. So जैसे इसका phagocytosis function है, वैसे ही इसका भी function phagocytosis है. I am going to write down over here function is phagocytosis. Okay. Now this and this phagocytosis is slightly different. ये क्या करेगा? ये भी फॉरेन माइक्रो ऑर्गेनिज्म को खा जाएगा, बट ये तब खाएगा जब देयर इज एन एंटीजन एंटीबॉडी कॉम्प्लेक्स. सो देयर इज स्लाइट स्पेस ओवर हियर. यहाँ पे ध्यान दो, नाउ दिस एंटीजन एंटीबॉडी वी विल बी लर्निंग इन अ सेपरेट चैप्टर. बट एंटीजन क्या है, एंटीबॉडी क्या है? सिंपल वर्ड्स में देख लो यहाँ पे. दिस इज योर फॉरेन बैक्टीरिया and these are the structures which are present on foreign bacteria. These are called antigen. ये जो structures दिख रहे हैं. So antigen is the foreign body. और आपके जो बॉडी के डिफेंस सेल्स है, उनके जो स्ट्रक्चर्स है जो इसके ऊपर अटैच हो जाएंगे, these structures are called as antibody. So your body produces these antibodies which attach to the antigen. This complex is called antigen antibody complex. Clear है? तो जैसे ही ये फॉर्म हो जाएगा, बाद में ये सेल्स ये पूरे स्ट्रक्चर को खा जाएंगे. सो phagocytosis is done. The next point is that they also produce antitoxins. Antitoxins मतलब डिटॉक्सिफिकेशन के लिए काम आता है. टॉक्सिन्स निकाल देते हैं, उसके लिए जो केमिकल्स है, they are called as antitoxins. Is this clear? And one more thing, see, during allergy what will happen is ये ज्यादा रिलीज होंगे. If basophils are released in more quantities, so obviously allergic condition means histamine will also be more. If histamine is more, what will happen is, अभी histamine बहुत ज्यादा भी बढ़ जाता है तो it is harmful for the body. So these cells are antihistaminic. See, there are so many words you have to remember. I will write it over here. Antihistaminic means that when the histamine levels go high, these cells will lower the histamine level, so it is called antihistaminic. So ये भी cells तब रिलीज होते हैं एलर्जिक कंडीशन में, लेकिन क्यों? क्योंकि बेसोफिल numbers are very very high. So if too much histamine is released, it can give a bad reaction to the body, and to stop that excessive histamine from being formed, these eosinophils they reduce the histamine. So these are also produced during allergy, but पहले ये होते हैं, बाद में ये होते हैं. So the third function I will write down anti-allergic. Okay, so देखो आपको शायद पता होगा there is a medicine called as cetirizine when you have cold. So cold के टाइम पे हिस्टामिन बढ़ जाता है, तो cetirizine is antihistaminic, वो ये आपके बॉडी में कम करेगा. So your mucus, your blocked nose is because of histamine. So when you take that medicine, आपका blocked nose कम हो जाएगा, mucus कम हो जाएगा. Why? Because it is antihistaminic, it will reduce the histamine. Same thing these things are doing. Is this clear? So we have finished with eosinophil. So you have the diagrams of all the different types of granulocytes. You have their properties, proportion, very very important to remember. And with this we have finished with granulocytes. Granulocytes I will teach you in the next lecture. Again ये theoretical topic है, so please ये 2-3 बार आपको पढ़ना है. So I hope everything is clear, and next lecture we will be completing with granulocytes, and we will be completing with last type of cells called as thrombocytes. Okay, so उतना ही आज के लिए. Thanks for watching.